Okay, so now we'll go on to the next speaker, and his title is very appropriate because it's reframing the question. So now we're going from populations down to the biochemistry of the bacteria and seeing how that affects metabolism and susceptibility. So Matt Redinbo.
So thank you, Helmut, and thanks to the committee for inviting me to participate in this meeting. And so as Helmut said, I was asked to talk about reframing or attempting to frame questions about the biochemistry of the microbiome toward understanding its impacts on not just environmental exposure, but on human health in general. And so that's what I'm going to try to do today. And what I thought I would do is really frame the questions towards understanding and then modulating validated biochemical pathways in the GI microbiome. And to me, that's both understanding as well as modulating them is really part and parcel to what I think we can do now and what we should be thinking about doing as the future evolves in terms of this field. And so the GI microbiota, as everybody here knows, is a very vast and dynamic repository of really the most amazing chemistry on this planet, not just in the GI, but as everybody knows, the bacteria are the greatest chemists we have, and the more we can learn from them, the better off we will be. And so, of course, with respect to the GI, these microbes impact a lot of physiological states. You could argue almost every physiological state is impacted by this vast extra organ that sits within us. And as it was alluded to by Andrew, we now have the technology, and as you all know, to really probe this for the first time and shine light on this organ in a level of granularity that I think is really revolutionary. And so from my point of view and from the point of view of this, this conference, the thing I'm most interested in is chemical toxicity and how the GI microbiota can respond to and contribute to and be modulated with respect to the toxicity that either endogenous or exogenous chemicals can provide. So what I've done here is I've pulled a series of chemicals from the 13th report from the NIEHS on carcinogens. And so this is just 24 of them. There's dozens and dozens in this report. And as I'll show in a little bit, I chose these for a good reason, but these include some very famous ones, the BAP, which is a polyaromatic hydrocarbon, as well as the polycyclic aromatic amines that are in cooked meat, as well as many others that are listed up here. And so this, as a chemist and a biochemist, this is really how I think about toxicology. I start with the chemistry of the situation, and then I attempt to sort of walk that forward to understand how the system can respond. And so that's what I thought I would do in this case is think about the elimination, the processing and elimination of these compounds first in an anthrocentric point of view and a mammalian-centric point of view. How do we respond to these types of chemicals? And so as everybody here knows, also we have a very well-established understanding or framework that we put on xenobiotic metabolism, and it goes really in 3 phases, but I'm going to show only 2 here. And the first phase is functionalization. So you take a nasty chemical and you attempt to introduce some functional groups to it. So our big heroes here are the cytochrome P450s that really are oxygen-driven blowtorches that can put oxygens in different places on molecules, really working uphill, as well as many other types of hydrolases and oxidoreductases. The second phase of drug metabolism in us is conjugation. So at this stage, you take those functionalized molecules and you add a conjugative group to it. The one I've shown here is glucuronic acid. You can also conjugate with sulfate, with amino acids like glycine, as well as with glutathione. And so this is really unique to us because this is the mark that we put on chemicals that tell the system to send these chemicals out for excretion. And that's harder for us because we have many tissues. So if the liver does this and decides it needs to send something out, it can't just modulate it and then hope that it'll dissolve out and find its way into the bloodstream and the kidneys. These marks tell it that this really needs to go. And they're sent out in a very organized way from the liver, typically through the bile, and they end up in the GI. So we'll sort of hold that there. And so then what I did for this talk is I did something that probably is well done in the literature, but I'm a simple-minded guy, so I did it for myself. And I just said from an anthropomorphic or anthrocentric point of view, do the bacteria contain these types of pathways? So this is our best understanding of the pathways that xenobiotic metabolism has. And so I asked, do the bugs have these? And so I listed just a series of enzymes in this phase 1 or phase 2 drug metabolism process, ones that are in mice and men. And then I just went into the HMP and the GI listing of genes, and I just looked for corresponding gene products. And what I found is for phase 1, for the functionalization enzymes, there were always orthologs that were present. Very good E-values, ones I could definitely put a green check mark next to. But perhaps not surprisingly, given what I just said, the phase 2 enzymes are missing from the microbiota. There's a few that look close in terms of percent identity or similarity, but really if you look at the E-values and you look for their catalytic residues, you find that they just aren't there. So the bacteria can do some amazing things, but what they don't do is conjugate things and send them out in that way. So you can ask yourself, well, so let's just summarize this. So they can do the types of chemistry that phase 1 enzymes can do, but they can't do the types of chemistry that our phase 2 enzymes can do. So if you set it up that way, you can then ask, well, what do the bacteria do? And this is the biggest intellectual punt that I have in this talk. There's a looming and vast et cetera that's on the bottom left part of the center column here. And so I pulled just a small number of types of enzymes that are known to be in the GI microbiota in terms of xenobiotic metabolism type enzymes. And there's a few listed here. But what we really need to appreciate is that because they're great chemists, they have to contend with a great diversity of chemistry. And so they have a really an astonishingly large swath of types of enzymes that we really need to go in and examine and understand them. And in addition, the molecules they make, as was alluded to again by Andrew, go across and interact with our tissues and activate PXR and FXR and the aryl hydrocarbon receptor. So their products have a huge impact on even our drug metabolism responses, right locally, as well as systemic responses. And so we really are beginning to understand this, and one of my favorite examples is from Peter Turnbaugh's lab, and this is the inactivation of digoxin, which he drove down, and I'm a reductionist at heart, and so he drove it down to a single species and a single gene product in that species that performs this inactivation, oxidation, reduction pathway. So there's a great deal to be discovered here, and hopefully we will fill in this et cetera as we go along. But because this is still relatively uncharacterized, I thought I'd go back to this question of the phase 2 in terms of how we do it. We conjugate things and we send them out. So is there an orthologous or paralogous pathway that the bugs do? And it turns out there is, and it's the reactivation of these compounds by many different types of enzymes expressed in the GI microbiota, as well as other places. And so this is really crucial because the reactivation is the word that I really want to drive home. These are compounds, when we send them out, they are almost always inactive. So by conjugating them with something, they no longer have the biological activity that they originally had. They're safe. After the bacteria finish with them, however, they are reactivated back to a form that could have very potent activity. And as I'll allude to, this can be very critical. So the yin to our yang is their reactivation by deconjugating the things we send out in a conjugated form. So I'll just give two examples. So cholic acid and many other bile acids are inactivated in many ways. And in one way, they are conjugated to amino acids. And in this case, I've showed cholic acid being fused to glycine to make glycocholic acid. And that's what we do. And then the bacteria see this, and they encode a whole series of bile salt hydrolases that can reverse this. And so cholic acid is pretty inert, but lithocholic acid or deoxycholic acid, these are very biologically active compounds and can be very toxic to our tissues. So this reactivation can be quite negative. And in the system that I'm going to talk about for the rest of the talk is the reactivation of the inactivated glucuronides, which are acted upon. So the glucuronides are made by a series of enzymes that take this really unique sugar and stick it onto things. And when that hits the GI, the bacteria see, they're just like my kids. They see this sugar, and they have no choice but to evolve systems to acquire the sugar as efficiently as possible. And so they've done that, and this is a carbon source. So this is a very efficient way to get energy. And so they take these inactivated glucuronides off, and they reactivate the compound. So in this case, I've shown diclofenac, which is an NSAID, and I'll come back to that a little bit later. This is a compound that we know now has severe GI tox, and I'll go into that in more detail. Okay, so the reason I picked the 24 compounds that I showed earlier was that all 24 of those known or almost certainly known carcinogens are inactivated via glucuronidation. And they almost certainly reach the GI in this form, and then we hypothesize would be subject to reactivation by the vast different types of beta-glucuronidases encoded by the GI microbiota. And so it turns out there are 11 known carcinogens that are glucuronidated, and there are 35 highly suspected or almost certainly known carcinogens. And so this is how I really want to frame, if you really talk about reframing a question, this was the question my lab really framed a few years ago, which was, could you go in and control what the microbiota are doing, but in a non-lethal fashion? And the non-lethality is critical because currently all things that attack bacteria that are clinical drugs are antibiotics, every one of them. And so, I don't have to tell this audience or any of the people here who have read the literature, we don't want to eliminate our bacteria. And so we want to go in and treat the bacteria in a way that allows you to modulate their behavior without wiping them out. And in that way, what we really are thinking about is that the millions of genes that are encoded by the microbiome contain therapeutic drug targets, and ones that we can attack in the same way that starting with antibiotics so many years ago, which were targeted compounds, we then understood what their targets were. We could then use that target information to design better drugs. So in my mind, the antibiotics really created the field of drug discovery. We can now turn that field back to the bacteria and say, let's find enzyme targets in the bacteria and treat them in a non-lethal way and shut down unwanted enzyme activities. And so the reason we did this was for a chemical toxin that we actually give to people. And that's an anti-cancer drug called irinotecan. So this is the activation and inactivation of irinotecan. It's given to patients as a prodrug. Through phase 1 drug metabolism, it's activated to its active metabolite, which then goes off and finds the tumor and poisons the activity of human topoisomerase 1, and that slows tumor growth. This is a very potent and effective compound. It's first line, or it's actually late stage for colorectal cancer, and is now first line for pancreas cancer. Pancreas cancer, you have to go in and hit that malignancy with as strong as drugs as you can. So the active component is then inactivated through glucuronidation. That marks it for excretion into the GI. And then in the GI, this compound gets reactivated. So this is pretty bad if this is lithocolic acid. This is really bad if this is an incredibly potent anti-neoplastic compound. And so what it does is it causes severe GI damage and dose-limiting diarrhea. And in single agent use, this is almost up to 90% in terms of all grades. And grade 3 or 4 diarrhea is almost hospitalization stage. So this is not something you want to experience. And so our idea was just to treat this toxicity as a drug discovery platform. In other words, if the bacterial beta-glucuronidases are the cause of the reactivation, then inhibitors to that set of enzymes should alleviate the toxicity. And so this was the overall goal, that if you could prevent the reactivation of the toxic metabolite in the GI, you could prevent the GI toxin dose-limiting diarrhea. So in setting up this goal, we put 3 rubrics behind it. The inhibitors we discovered had to be potent, so they could have on-target activity. They had to be selective, because it turns out you have a beta-glucuronidase as well, and it is required for degrading things that are brought to the lysosomes. And so if you have an inactivation of your beta-glucuronidase, you have a disease called Sly syndrome, which is pretty horrible, as are all lysosomal storage diseases. So we had to be selective. We had to have compounds that would only hit the bacterial enzymes. And then, as I said, we wanted to be non-lethal. Unfortunately, that was pretty easy. As anybody who's tried to discover antibiotics knows, it's actually pretty hard to kill bacteria. So we were quite fortunate there. So what we did was we took E. coli beta-glucuronidase and we performed a high-throughput screen looking for potent inhibitors. And what I show here is a set of 4 inhibitors that are all of a similar chemotype that were potent inhibitors. And what I'll tell you is that we have dozens more, and getting inhibitors for this particular enzyme wasn't hard, fortunately. But I'll walk you through just this chemotype in this presentation. And so the first thing we were very fortunate was that they were all potent. So we had, straight from hits, we had double to triple digit nanomolar potency. And so we're a structural biology lab as well as a couple other things. And so we solved the crystal structures of the target enzyme in complex with our hits. And in doing this, we identified a key interaction that, again, was very fortunate for us. So the inhibitors bind to a loop that I've turned here, the bacterial loop, that is a feature of the bacterial enzymes that's missing from the mammalian or human beta-glucuronidase. And so because our inhibitors target that loop, it turns out they're very specific, completely specific, 10 to the 5th specific. So I was quoted in C&E News a few months ago as saying that we were superhuman lucky, which is great because I spent about 45 minutes talking to this reporter, and that was the one quote that she picked up. And now appears in many places around my laboratory as the students all say, hello, Mr. Superhuman Lucky. So we certainly were, and you have to be fortunate. And so we got compounds that were very selective. And I won't show you, but we also, all these compounds are non-lethal. So we managed to check off all 3 of our rubrics that we set up ahead of time. And so the last thing we wanted to check was whether or not they did the thing we really wanted them to do, which was prevent GI tox in animals treated with irinotecan. And so the results are shown here. So if you treat an animal with irinotecan in the upper left, sort of the center set of bar graphs, all the animals get bloody acute diarrhea after about 9 or 10 days of exposure. If you treat them with one of our inhibitors orally, twice daily, you significantly alleviate the onset of that toxicity and the diarrhea. And the histology, I think, is the most revealing. So the pictures on the left show either untreated or vehicle-treated with just our inhibitor, and that's a normal mouse colon. The third one over is what a mouse colon looks like that's been treated with irinotecan. So you see there's no glandular structure left. All the blue cells are inflammatory cells. So this has really destroyed this particular tissue. And then with our inhibitors on top, we see a protection and a more normal looking colon. So, of course, we've moved on from this, and one of the things we appreciated early on was that looking at E. coli as the only beta-glucuronidase was sort of like considering Justin Bieber to be normal in terms of humanity. And so we really had to design and figure out what this family of enzyme targets looked like. And so when you do drug discovery toward a human enzyme target, fortunately, usually you have one or just a few splice variants that you have to contend with. Here, we had to dive into the structure function of this enzyme. And really, the first thing we had to do, so these are a few of the structures we've solved and reported, but what we really had to do was be good chemists and look at the active site and pick out the key residues that would define the beta-glucuronidases relative to the many other sugar hydrolyzing enzymes that are in the GI, particularly the beta-galactosidases, which are very close. And so it turns out we identified some key fingerprint regions that really define what a beta-glucuronidase is. And in doing that, we're able now to go into the HMP GI database, and we are annotating now and about to report the Gussome, the entire catalog of Gusses that are in there. And it turns out there are 3,013, and you can reduce that down to 293 that are non-redundant. And they have a wide variety of features. So here's just a few examples that we've looked at. Those are the ones we've already reported. And then there's some bigger ones that begin to come out, and you see some different phylogeny here as well. And they even get a little bit bigger and then bigger than that. And in fact, they go up to about 950 amino acids in length. And what we found, and have yet to report, but I'll tell you, is that they have an incredibly vast set of differences in terms of the types of substrates they process. And this makes perfect sense. The GI is the great crowdsourcer of chemistry. They don't have to have a fixed genome. They can have an incredibly vast and dynamic genome, and different things can be expressed at different times. So if you eat a heavy meal and you secrete a lot of bile acids into your GI in millimolar quantities, you're probably going to get at least an upregulation, if not a slight bloom of species that encode the types of enzymes that can process bile acids. And so we consider this to be very exciting and a way to kind of examine what this Gussome does and figure out which substrates are processed by which enzymes. And I'll also tell you, just this set, as well as the other ones we've looked at, have a great difference in terms of their propensity for inhibition. So the compounds we've worked with to date are very good at some of these enzymes and have no effect on others. And so we see this as a way, if you bring this back to toxicity, if you want to prevent GI toxicity by chemical toxicants, we could potentially tell you which are the dangerous bugs and then potentially bring along regulators modulating compounds that can stop the activity of that particular type of beta-glucuronidase, or by extension, other GI enzyme. So I'll just run you through a quick set of additional data. So we moved on from irinotecan as a chemical toxin to the non-steroidal anti-inflammatory drugs. And so this is a little bit controversial. When I got into this, I thought what most people think, which is that your NSAID toxicity is upper GI, it's stomach. And it turns out with video capsule endoscopy, it's now known that long-term NSAID users, 70% of them have significant damage to their GI, 40% of them have malabsorption, and 30% of them have frank bleeding ulcers. And that's all in the lower GI. So until you could visualize that, clinicians and the scientific community didn't realize the damage we were causing. And so, as I alluded to before, diclofenac is glucuronidated, as well as all of the carboxylic acid containing NSAIDs, and they form acylglucuronides. So we went in a similar pose to irinotecan, and with diclofenac, we treated mice with an ulcerogenic dose of diclofenac, and that's shown in the center. So there's a longitudinal ulcer there. And then if you treat with our inhibitors orally at the same time as the ulcerogenic dose of the NSAID, you can significantly prevent that ulcer toxicity. And so we've done that also with a second NSAID, endomethacin, and so that's shown here, and then with a third ketoprofen. And so we've attempted to bring to the forefront the idea that you can target GI bacteria toward the goal of modulating the damage that metabolites of specific drugs can have on the GI. Now, for a clinical pose, I have started a little startup company whose goal is to try to bring forward some of these inhibitors toward preclinical leads with the goal of alleviating the toxicity of irinotecan. And so we're in the midst of that, and whoops, there's our company's name, and fortunately we are making progress. But I also happen to know that there's a much bigger opportunity here. And again, I'm just going to stay focused on the Gusses for the next few seconds, and then I'll expand it a little bit. So just from the point of view of the beta-glucuronidases, the Gusses, remember all of these toxic compounds, and I would hypothesize that the potential is to go in and alleviate the toxicity that's caused by these type of chemicals by modulating this same family of enzymes in the GI bacteria. And so the way I think about this is that we have found a needle, which is the beta-glucuronidases, and there are about 3,000 orthologs in the GI. But there's many more to be found in the 2 million or 3 million or 20 million, whatever the total number of genes in the GI microbiome. And so the last thing I'll say in terms of framing is I thought I'd just take you through the way we frame these types of questions for ourselves in the lab and in the way we set up our experiments. And so again, to be a reductionist chemist, you have to understand the detailed chemistry. And to me, that's so important because you can do a lot of associative things, but unless you really understand, what is the cause of the problem, how is that cause modulated and impacted by all the systems you're looking at. And so we really go to that point. And when we have a strong feeling on this first bullet point, we then move ahead. And so in doing that, you identify candidate genes, and then you have to establish what I call really a cycle of well-characterized and robust in vitro systems that you can use to examine your adjuvant, your set of enzymes, and its effect. And if you can work that out in vitro, that is so powerful, because then you can take that and you can move into an ex vivo mode where you can take fecal material or cell-free fecal extracts, and you can ask and answer the same types of questions. You can also culture on differentiated colon or small intestinal cultures. You can actually, if you're clever, and hopefully we're working with somebody who's quite clever to do this, you can culture those and have the human cells, the mammalian cells, be aerobic and put an anaerobic layer on top of that of bacteria. And then you have a true ex vivo potential model of what's going on. And to me, that is really critical, because notobiotics and going into an in vivo mode, which I have up here and we always do, and we do this in a cycle, but going into a mouse or a notobiotic mouse is costly and time consuming. And so, but of course, it's the only way you can do the other things that I think are crucial, which is you have to go in and do sequencing, you have to do metatranscriptomics if you can afford it, and you have to be able to do metabolomics and really look at the chemical effects of this system. And so this is the way we've set this up in our laboratory and in our work with collaborators. Okay, so I'll just end by thanking all the people that did this work, and my critical collaborators here were Shrit Harmani at Einstein College of Medicine, and Urs Bolsterli, who was at UConn and has since retired. And I thank all the people in my lab, past and present. And thank you very much for your attention.
Marty Blazer, NYU. Matt, beautiful, really outstanding work that's going to be terrific for decades to come. So I wanted to ask you about the idea of looking for agents that are non-lethal, and presumably that's because you want to decrease selection for resistance. Mm-hmm. But even if they're non-lethal, there can be selection for resistance. And my question, because you're depriving bacteria of a foodstuff, and mm-hmm. a variant may arise that that is resisted and obviously has a selective advantage. So have you found escape mutants yet? So
that's a great question. We have not gotten to that point where we've looked for them. We've banked everything, and that particular question has not arisen in terms of what we want to dive into sequencing. I'd like to do that. In my mind, though, I'd like to first understand more of the dynamics of what happens in a normal mouse when you give one of our inhibitors and you just say, you know, what happens to the GI microbiota? There's going to be compositional changes almost certainly. I would hypothesize the changes would be robust and recoverable. In other words, if you take the inhibitor away, I would hypothesize that the bacteria would probably return back to their state. I'd like to understand that first, and I don't understand that yet. And then, under a situation of maybe chronic exposure, we would then sample and look for mutants. I'll give you my hypothesis, though. It's unlikely we're going to get them, because I think the system is very dynamic. And the bugs that have one that could mutate, they'll just be de-repressed or be repressed, and they'll just fall back and won't really bloom. So there won't be that much selective pressure on them. Another way to do this would be to do it in vitro and just really push the system and see what we get. And I like that, but we haven't gotten to that yet.
You know, I just want to push back a little, and that is that in the short term, probably it's not going to be a problem. But if you're looking at these as drugs for chronic conditions, that will be the opportunity for the emergence of resistance, which may limit the utility. But
yeah, absolutely.
You know, chances are we're going to get there because nature is infinitely knowledgeable.
Yeah.
Ann Summers, Georgia. I love the superhuman lucky business and also the Gussome. Those are great. I have two quick questions. One of them is a chemical question and one of them is a microbe question. The chemical question has to do with whether or not, and this has to do a little bit with what Marty was asking about, and the subject which I'm concerned greatly about, which is antibiotic resistance, is did you at any point, or do you think it would be to the point, given the great variety of these things out there, to profile which, when you've got a target, a therapeutic that you don't want inactivated, the irinotecan, okay, to ask either in the lab or computationally, which one of these various gusses that are out there would actually be the one that you want to hit because it's messing with this guy. So that's the chemical question. And then the microbe question is, that was a very specific one. Have you used in your ex vivo model the altered Shadler flora? And what do you think about doing that for ex vivo models? Thanks.
So two great questions. So the second question is, we know of the ASF bugs, and we have, I think, 5 of the 8, and we haven't yet been able to identify the other 3. We've wanted to use those, and probably will at some point within one of our studies, in the midst of a mono-associated, these 8 associated, and then a complex associated, and map those differences. The question about which gusses are the most crucial, we'll publish this shortly, but I'll just tell you, the proteobacterial ones are the most active. And this is now becoming a pretty common theme, that E. coli and its cousins really shouldn't be in the GI, but is. And where they're there and the opportunity provides them a good chance to kick ass, they do. And they do it in a bad way. And so that's what we've seen to date, and it's really interesting because they have only the proteobacteria have a very organized operon that will recognize a glucuronide, has a transcriptional repressor that de-represses the entire set of genes, including transporters, which actively pump in these glucuronides. So these guys have concealed weapons, and they are ready to shoot in terms of going after these things. And so if you give them a strong glucuronide source, they bloom up. And so that's our current hypothesis, and that's similar to the LPS story that we've seen in terms of colitis and colorectal cancer. They shouldn't be there, but when they're there, they make bad stuff. It's
again, this goes back to the theme that Marty raised. How often is that Gus locus on a mobile element?
Yeah.
They call them proteobacteria for a reason.
Yeah, so I have great love, and maybe a little bit like Maria, I had to give up on my love of studying the structure function of antibiotic resistance propagation because nobody would return my funding calls anymore. And I'd love to get back to that. I should have looked at that because I spend a lot of time looking at mobile plasmids. I've never looked for it, but I've never bumped into it. So it'd be interesting. I'll go and take a look at that.
Betcha.
And that's the last question. So can you flip this around and instead of inactivating enzymes that are detrimental, can you find drugs that allosterically activate beneficial enzymes?
Yeah, I think we can. And I think the mirroring with the FXR, our ability to respond to compounds that will upregulate certain things, certainly those pathways exist in the bacteria. They're not the same, obviously. They don't have PXR and FXR, but they do have things that will recognize specific types of positive regulators, and we can use those as a feed-forward mechanism. Now, again, I'm very much of a reductionist. I don't have a specific set of genes that I know I'd want to or can regulate, but I think if we find one of those, we'd be excited to try to move forward with that.
Now, I believe, right? When do we come back? I don't know my schedule. Anyways, you have your schedule, so be back on time. 10:45, thank you.